Hello everyone, this is your educator Priyanka Thakur teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. So this is your photosynthesis part 5, but you're not watching my videos, but it's alright, it's all your choice. I'm not forcing you to do so. Well, but in part 5, we are going to discuss about our C2 cycle and C4 cycle, also known as photorespiration, glycolate cycle, and Hatch-Slack cycle, respectively. Okay, in the next video, I think it's gonna be the last part of photosynthesis. In that particular part, you will got to know about difference between the C3, C4 and CAM cycles. Along with that, you will be solving some of the MCQs, so stay tuned. Okay, so without wasting our time, let's start with our today's topic, which is a C2 cycle first and then C4 cycle. Okay, here is our C2 cycle and also known as photorespiration or glycolate cycle. Fine. So, in what kind of plants we can see C2 cycle? It mostly occurs in the C3 plants. In C3 plants, we can see Calvin cycle, C3 cycle as well as C2 cycle. Why C3 cycle is called so? Because the first stable product in case of Calvin cycle is phosphoglyceric acid, which is a 3-carbon compound, and that's why Calvin cycle is also known as C3 cycle. But here we are going to talk about C2 cycle, which is also known as photorespiration. Okay, so what happened in C3 plants? We know that it is known as C3 cycle. Along with that, those plants are also known as C3 plants. Fine. So in C3 plant, what happens? Again, we know that ribulose 1,5-bisphosphate is the first acceptor of carbon dioxide in C3 cycle. But here, in case of photorespiration, what happened? When the carbon dioxide concentration decreases in the atmosphere, oxygen concentration increases due to the increase in temperature. This rubisco starts acting as oxygenase instead of carboxylase. You know that rubisco is a bifunctional enzyme. It can act as carboxylase as well as oxygenase, but it acts as oxygenase when the temperature is high, light intensity is high, and oxygen concentration is high and carbon dioxide is low. It will be acting as oxygenase. So now instead of fixing the carbon dioxide, it start fixing oxygen. You know, photorespiration is known as a wasteful process. Exactly, it is not a wasteful process. Clearly, I want to tell you. You will see that there will be the formation of sugar, but still, it is a very tedious process, and it is a wasteful yet not wasteful process, I can say. Fine. See what happened. Ribulose 1,5-bisphosphate, which is a 5-carbon compound, gets oxygen gets incorporated in it instead of carbon dioxide. So we still have 5-carbon compound. There is no formation of any 6-carbon compound. Instead, phosphoglycolate, which is a 2-carbon compound, and phosphoglyceric acid, which is a 3-carbon compound, are formed. So one is phosphoglycolate, other is phosphoglyceric acid. Clear? And here is the enzyme rubisco acting. Okay, it is acting as oxygenase. Clear? Okay, we got to know it. So phosphoglyceric acid now will get involved in the process of formation of carbohydrates. It will be used up during the C3 cycle, but this phosphoglycolate will not be metabolized. Okay, it will be metabolized in a different way. For photorespiration, we are having 3 compartments: chloroplast, peroxisome, and mitochondrion. Okay, repeat: chloroplast, peroxisome, and mitochondrion. Why am I asking you to repeat? Because many times you forget about the compartments. So even, okay, the first is chloroplast, second one is peroxisome, third one is mitochondrion. Okay, Phosphoglycolate is not metabolized here, and then one inorganic phosphate is removed and phosphoglycolate is converted into glycolate now. This glycolate formed in the chloroplast now move to peroxisome. This is glycolate 2 carbon compound, and this C2 cycle is also known as glycolate cycle. Why C2? 2 carbon compound it is. Okay. Now this glycolate formed in the chloroplast will move to the peroxisome. Great. Okay. It now moves to peroxisome. Clear hai? Is it clear? Okay. Then here come our marker enzyme, oxidase. Glycolate oxidase is known as the marker enzyme of photorespiration. Glycolate will be converted into glyoxylate with the release of hydrogen peroxide. Okay, you know what is special about, unique about this photorespiration? The conversion of phosphoglycolate into glycine, and there is one step, decarboxylation. Clear? See, we got to know glycolate, which is formed in the chloroplast, will get transferred to peroxisome. Here oxidase marker enzyme of photorespiration is found in peroxisome, not chloroplast, in the peroxisome. So glycolate will be converted into glyoxylate with the release of hydrogen peroxide. Now glyoxylate will get converted into glycine, where glycine is formed in the peroxisome. Fine. Glycine will then move to mitochondrion. There will be 2 glycine molecules rather than 1. Okay, you can see transamination reaction here. But let's talk about it later. Okay, glycine moved to mitochondrion. Glycine, you can see there is no decarboxylation. Glycolate is a 2 carbon compound. Next, glyoxylate will be having 2 carbon. Glycine is a 2 carbon amino acid which will move to mitochondrion, but there will be 2 molecules of glycine. So we are having 4 glycine here. How many? 4 glycine here. Okay. So serine is a 3 carbon amino acid. There will be 1 carbon dioxide which will be released. Carbon dioxide, there is decarboxylation. Carbon dioxide will be released along with this, as there are 2 molecules of glycine. 1 amino group will be released. NH3 will be released. Okay, keep in your mind where NH3 is released, where carbon dioxide is released. Okay, in the mitochondrion. So here NAD positive gets converted into NADH, and when glycine is converted into the serine, at that time you see the decarboxylation process and the release of NH3 as well. Carbon dioxide, which is needed to be fixed during the Calvin cycle, is being unfixed during the photorespiration. Okay, this is one of the biggest disadvantage of this photorespiration. Okay, carbon dioxide is released to outside. This serine moved to peroxisome. Serine is formed in mitochondrion. Such kind of questions are usually asked, that where serine is being formed, where glycine is being formed. Okay, this serine move to peroxisome, where it gets converted into hydroxypyruvate. There will be conversion of NADH into NAD. NADH will be used, and hydroxypyruvate will be converted into glycerate. Now glycerate is formed in peroxisome. Glycine is formed in peroxisome. Okay, glyoxylate is formed in peroxisome. Marker enzyme of photorespiration occurs in peroxisome. Clear or not? Fine. And this glycerate will move to chloroplast now, where it requires a phosphate group to get converted into the phosphoglycerate. This glyceric acid will be now converting into the phosphoglyceric acid, and it requires a phosphate group which will be provided by the ATP. ATP will be utilized and it is converted into ADP, and glyceric acid now get a phosphate group, and this phosphoglyceric acid now takes part in the C3 cycle. Okay, in C2 cycle, just like C3 cycle, IUPP is the first acceptor, but instead of accepting the carbon dioxide, it is accepting oxygen here. Okay, so there is no there is one phosphoglyceric acid. In case of C3 cycle, there are 2 molecules of phosphoglyceric acid. Do not forget it. Do not forget anything. You know, you should remember, for 1 carbon dioxide, there is formation of 2 phosphoglyceric acid in case of C3 cycle. Here there is formation of only one phosphoglyceric acid, okay, and one is phosphoglycolate, which is metabolized in this manner now, okay. And this process, definitely there is phosphoglyceric acid which can be utilized in the C3 cycle, but still there is a wastage of energy. There is unfixing of carbon dioxide. So isn't it a wasteful process? It will result in the decreased productivity of the plants, okay. If C3 plants, when they get a higher light intensity, they are present in the more temperature, they will be performing photorespiration rather than Calvin cycle. So this process can decrease the productivity of C3 plants. So they do have to think about something else. They have to adapt according to their environment so that they can increase their productivity. You understand? Our C3 plants need to increase their productivity. So in order to increase their productivity, what they need to do, they need to think of something else, because this photorespiration is decreasing their productivity by wasting their energy while unfixing the carbon dioxide. Now what I told you, what you have to remember about the marker enzyme, about the glycine formation, about the serine formation, about the glycerate formation as well. Okay, clear? Let's move to our next topic now. C2 cycle completes here. And here is Kranz Anatomy now. C4 plants. In C4 plants, you will see a special type of anatomy, Kranz Anatomy. Now, what happens in this Kranz Anatomy? In Kranz Anatomy, what we see, there is two different types of cells. One is bundle seed cell having chloroplast, another is mesophyll cells with chloroplast. Okay, one is bundle sheath cell, other is mesophyll cell. Okay, this bundle sheath cells have large chloroplast, while mesophyll cells are with a few and small chloroplasts. Other than this, the chloroplast in a case of bundle sheath cell is agranal, while in case of mesophyll cell it is granal. Okay, mesophyll cells are having granal chloroplast, and bundle sheath cell has agranal chloroplast. What happens in agranal chloroplast? In agranal chloroplast, there is no formation of grana. We cannot see the grana. Instead, we see only lamellar type of formations. Okay. So that's why the chloroplast in case of bundle sheath cell is known as agranal chloroplast. So now the Calvin cycle is going to be there in bundle sheath cells and not mesophyll cells. Let's talk about it later. You got to know what happens in case of Kranz Anatomy. There is our bundle sheath cells, so having large chloroplast, agranal chloroplast, and there is the formation of a wreath. In wreath-like manner, there is the formation of mesophyll cell. Wreath means in ring-like manner. This is what we call Kranz Anatomy. Okay, this is a wreath formation of mesophyll cell in ring-like formation of mesophyll cell. Okay, clear about Kranz Anatomy? Large chloroplasts are found in bundle sheath cells. Small chloroplasts are found in mesophyll sheath cell and mesophyll cells. Okay, which one is toward outside? It's mesophyll cell, and inside it's bundle sheath cell. Which one is having agranal chloroplast? Bundle sheath cell. Great. Okay, you understand it now. So let's talk about our C4 photosynthetic pathway. What exactly happens during the photosynthetic pathways? See, here atmospheric carbon dioxide is being fixed. First acceptor in case of C4 photosynthetic pathways, phosphoenol pyruvate. Which one? Phosphoenol pyruvate, which is the first carbon dioxide acceptor in case of C3 cycle. It's ribulose 1,5-bisphosphate, and in case of C4 it's phosphoenol pyruvate. Great. Phosphoenol pyruvate will be acted upon by PEP carboxylase. Actually, this carbon dioxide get converted into hydrogen bicarbonates. There will be a water molecule also in the presence of carbonic anhydrase. It will be converted into bicarbonates. So, it will be acted upon by PEP carboxylase. PEP carboxylase will incorporate the carbon dioxide into phosphoenol pyruvate. Phosphoenol pyruvate is a 3 carbon compound and RUBP is a 5 carbon compound. Okay, so here after the carbon dioxide has been incorporated into the phosphoenol pyruvate in the presence of PEP carboxylase, it gets converted into oxaloacetate, which is a 4 carbon compound. So the first stable product in case of a C4 cycle is the 4 carbon atom. That's why we call it C4 cycle, and the plants in which C4 cycle is occurring, it is taking place. Okay, these are known as C4 plants. So a first stable product in C4 plant is oxaloacetic acid having 4 carbons. In case of C3 plants, it is what it is. What is it? It is phosphoglyceric acid having 3 carbon. Okay. Oxaloacetate get converted into malate in the presence of malate dehydrogenase, and NADPH is used up here and get converted into NAD positive. Okay. Okay. Now this malate which is formed in the mesophyll cell will now move to bundle sheath cell. This mesophyll cell having malate, and malate in the mesophyll cell will now move to bundle sheath cell. Clear? Now bundle sheath cells, what happens in bundle sheath cell? This malate that enters the bundle sheath cells undergo decarboxylation reaction in the presence of malic enzyme, and then there is formation of NADPH. NAD positive will be converted into NADPH. One is used here, another one is formed here. Okay. So there will be the release of carbon dioxide also. So this 4 carbon compound will now be converted into some 3 carbon compound because 1 carbon dioxide has been released. This carbon dioxide that get concentrated in the bundle sheath cell and is now used in the Calvin cycle, and that Calvin cycle will lead to the formation of carbohydrates. As I have told you in my previous video that when you have read about phosphoglyceraldehyde in Calvin cycle, when phosphoglyceraldehyde goes into the stroma, it is converted into starch. If phosphoglyceraldehyde and goes into the it diffuse out of the stroma into the cytosol, it gets converted into sucrose. Well, other than this, okay, this is Calvin cycle, and this malate is now converted into 3 carbon compound, which is pyruvate. Okay, pyruvate feels like nobody is valuing me here, nobody wants me here. Okay, I should leave from here because nobody is asking me, nobody is valuing me, so I need to leave here. They only value carbon dioxide. This Calvin cycle values only carbon dioxide and not me. So what should I do here? I should move out of this bundle sheath cell. This is not a place for me to live. Okay, I'm going to leave you, bundle sheath cell. So this pyruvate will be leaving the bundle sheath cell and went back to the mesophyll cell, where it gets converted into phosphoenol pyruvate. And here pyruvate diphosphate dikinase will play its role, which will use 2 ATPs. Okay, instead of 1, there is the use of 2 ATPs, which will get converted into the ADP. Okay, so 2 ATPs are used during the regeneration of phosphoenol pyruvate. For 1 carbon dioxide, 2 ATPs are used for the regeneration of phosphoenol pyruvate. In case of RUBP, for 1 ATP is used. Here 2 ATP are used for the regeneration of phosphoenol pyruvate. So pyruvate in the presence of pyruvate phosphate dikinase, which will be using 2 ATP, gets converted into phosphoenol pyruvate. So for 1 carbon dioxide, there is the use of 2 ATP, and we know that to make 1 molecule, sugar molecule, glucose molecule, we require 6 cycles. So 6 carbon dioxide will be used. Okay, there is another cycle, Calvin cycle. So in case of C4 plant, total how many ATPs are used? 12 are going to be used here during the regeneration of phosphoenol pyruvate, and we already know that 18 ATPs are used during the Calvin cycle. You understand? 18 ATP are used during the Calvin cycle and 12 are used during the regeneration of phosphoenol pyruvate. So we exactly are having total 30 ATPs which are being used during the C4 photosynthetic pathway. How many ATPs are used in C4 photosynthetic pathway? There are 30 ATPs which are used during C4 photosynthetic pathway. Is everything clear to you? We know that the first acceptor is phosphoenol pyruvate, which will be acted upon by phosphoenol pyruvate carboxylase, which incorporate carbon dioxide in the phosphoenol pyruvate, which is a 3-carbon compound. It will be converted into the oxaloacetate, which is a 4-carbon compound now, which will be acted upon by malate dehydrogenase, and this oxaloacetate get converted into malate. There is no release of carbon dioxide. NADPH is being used. This malate get transferred to our bundle sheet cell, where in the presence of malic enzyme it undergoes decarboxylation. One carbon dioxide is released, NADPH is formed. Okay, this become equals. Okay, so 12 NADPH are formed, fine, are used as are used in Calvin cycle only, but total ATP are 30. Okay, and then the pyruvate is formed. Pyruvate went back to mesophyll cell, where using 2 ATP it get converted into phosphoenol pyruvate. This is what happened during C4 photosynthetic pathway. Now we know that PAP carboxylase is a carboxylase. It's not an oxygenase. Now it doesn't matter if the oxygen concentration in the atmosphere is more than the carbon dioxide concentration, because this PAP carboxylase is a carboxylase, for God's sake. It's not an oxygenase. So why it would be fixing the oxygen instead of carbon dioxide? It loves carbon dioxide. It is having the affinity for carbon dioxide, so it fixes only carbon dioxide, which is then released into the bundle sheet cell. And enzymes of Calvin cycle in case of C4 plants are found in the bundle sheet cells. Where? In bundle sheet cell. Is everything clear to you? Very good. That's great. Now you know about how many ATPs are used in the C4 cycle. Now you know where malic acid is being formed. In fact, you already know about many of things. But is everything to you is clear now? Great. I wanted it. Okay. So here we complete our C2 and C4 cycles. In the next photosynthetic part 5, we'll talk about CAM cycles, difference between all these. Along with this, we will be doing some questions related to photosynthesis, because we complete our photosynthesis, and after this we will be studying about hormones. Okay, so just wait and watch. We will be completing everything in detail actually. So that's all for today, but before leaving, okay, let me tell you that our Neha Taneja ma'am, beloved Neha Taneja ma'am, teaches on Unacademy platform. So if you want to study for if you are doing CSIR net preparation, you can follow Neha Taneja ma'am on Unacademy, and also you can see there are details of paid courses. You can check it out here. You can use the code Neha so that you will get 10% off for this. You will get a lot of access to a lot of classes there. So just if you want, if you are so much dedicated, go there, subscribe. Unacademy, I won't ask you to subscribe. Actually, you can, but go, on YouTube. Go subscribe there, as well as get the subscription of Unacademy on your platform, and follow our Neha Taneja ma'am there. That's all for today. Stay tuned and keep watching all the videos regularly without any absent. If you are doing so, that's great. Okay, that's all for today, and do not forget to subscribe to our channel, which is a small and lovely channel. It needs your love, so okay, give it some lots of love. Okay, subscribe our YouTube channel, join us on Telegram. But this is all for today. Do take care of yourself, and thank you.